Welcome, Homo sapiens. How are you? I am absolutely fine. Now, today in this video, I am going to teach you about tertiary structure of proteins. So, in this video, you are going to learn deeply about tertiary structure of proteins. Now, before going further, I request you, just have one glass water. Please have one glass water. Drink one glass water. Feel the coolness. Remember, this water is going to boost your memory. Keep in mind, the water is going to boost your memory. Okay? Just feel the coolness. And always keep in mind, you are 70% water. Now, here, hopefully, you have eaten well, you have played well, you have slept well, your body is feeling energy, you are feeling energy in your body, you are healthy. Okay? Now, let us just focus on your breath. Just focus on your breath. Slow down your breath. Slow down. Okay, just slow down your breath. Take deep breath. Slow down. Okay, take breath, hold the breath, release. Slow down. Very important. Now, just have your hand like this. Okay, just put your hand here. Just try to listen the sound, the most powerful sound. Okay, just try to listen. Try to focus your mind completely on the sound. Okay, just try to focus. Try to feel the vibrations. Okay, just try to feel these gravitational waves, marvelous waves, sounds. Okay, just try to feel. Okay, just calm your mind. Okay, don't think positive, negative, nothing. Okay, just listen carefully. Okay, today I am going to teach you about proteins tertiary structure. Just focus and listen carefully. Okay, today I am going to teach you tertiary structure of proteins. Now, here I just want to tell you, if you remember our previous classes related to structure of proteins, yes, we have discussed about primary structure. In primary structure of proteins, I have taught you about that primary structure is going to tell you about the primary sequence, the sequence of the amino acids which are present within the protein. Yes, it is also going to tell you about covalent bonds like disulfide linkages which are present within the proteins. Now, here if you see, we have learned in secondary structure. When we discussed about the secondary structure, what we learned that in secondary structure, generally we are going to learn about local repeating patterns which are being found within the proteins. In that class, we discussed about alpha helix, beta sheets, beta turns. Now, today we are going to discuss about the tertiary structure of the proteins. Keep in mind, the tertiary structure of the protein is actually the three-dimensional structure of the protein. So today we are going to have discussion related to tertiary structure of the proteins. Now here, I just want to tell you, here in tertiary structure, what we are going to discuss, the overall interaction between various amino acids. So hopefully you remember proteins, what are they? They are polymer of amino acids. So here in tertiary structure, we are going to discuss the overall structure interactions between various amino acids. Yes, here you are going to see interactions between folds, between domains. Don't worry, I will make you clear what are folds, what are domains. Now, here keep in mind in tertiary structure, okay, generally what you are going to see, a protein at the last is going to have unique structure. And here keep in mind this unique structure is responsible for specific function of the protein. You will learn here that if there are proteins which are going to have similar structure, they are also going to have similar function. So here the structure of the protein, it is going to decide the function. So here keep in mind why we want to study the structure of the protein, so that we can have idea about the function of the protein. Here I just want to tell you that if you see more than 1,50,000 protein structure, scientists they have studied, okay. And now if you are going to see various database like protein data bank, or you are going to have SCOP2, it is a database. Here if you see, it is structural classification of protein database. So here in these database, what you are going to see, the structure of various proteins are available. You can study easily. Now here if you see, to understand the 3D structure, we have to understand some terms. Like the first term you have to understand is motif, or you can say the fold. Now try to see here what is motif, what is fold. Here if you see, in a motif, what you are going to have, two or more elements of secondary structure, plus connection between them. So in a motif, what you are going to have, two or more elements of secondary structure plus the connection between them. It will be clear if you are going to see this picture. Now try to see here. Here in the blue color and the yellow color, okay, arrow, these are secondary structure. So here the blue one you are going to have helix, and here in the yellow arrow you are going to have sheets. So here you see these helix sheets, they have been joined through connections. So here this is going to represent one motif. So here what you are going to have, two or more elements of secondary structure. Okay, here you are going to have, okay, beta and alpha helix, one alpha helix and two beta loops or two beta structure, and they are being connected. Okay, so this structure is going to form beta alpha beta loop. Okay, beta alpha beta loop, this is motif. Hopefully it is clear what is motif. Now here if you see, if you are going to see various enzymes, okay, in various enzymes you are going to have common motif. Why? Try to see here. In enzymes you are going to have generally a repetition of beta alpha beta loop. So this secondary structure, it is being repeated multiple times, and this structure, okay, repetition of beta alpha beta loop, it is going to give rise a structure like this, and this is known as alpha by beta barrel. Alpha by beta barrel. Now here I also want to tell you the another term, that is domains. Try to see here, if you are going to have a large protein, in a large protein you are going to have multiple domains. So here try to understand what is domain. Here if you see, if you are going to have a large protein, you are going to have different globular lobes. Just for simplification, I am going to show you like this. So let's suppose there is a large protein and it is going to have these three different globular lobes. Yes, by using proteolytic enzymes, you can separate these lobes. The enzymes, proteolytic enzymes, they will cleave at specific position, and by using those proteolytic enzymes, you can separate these lobes. Now try to see here, these globular lobes, even though they are being separated, they still are going to maintain the same structure. Okay, now these different globular lobes, keep in mind, they are going to have different function. And they are going to maintain 3D structure. Now here, if you see, in the protein, okay, what you are going to see, if you see the proteins generally which are similar in sequence, they are also going to have similar 3D structure. The proteins which are similar in sequence, they are going to have similar 3D structure, and if the structure is similar, means they are going to have similar function. Here, such proteins, okay, which are going to have similar sequence, similar 3D structure, okay, similar function, they can be classified in a single protein family, in the same protein family. Now here, keep in mind, there are 4,000 different protein families. Now here, if you see, if you are going to have two or more families, and let us suppose if these families, they are going to have little similarity in amino acid sequence. Two or more families, and they are going to have little similarity in amino acid sequence, and if they are going to have same major structural motifs, okay, then they are also going to have similar function, because they are going to have same major structural motifs present in them. Okay, so two or more families. Okay, so there is one family, there is another family. Okay, and in between them, what is there? Little similarity in amino acid sequence. Okay, so such families, they can be grouped together in super families. Okay, so hopefully it is clear what are these super families. Now let us see one example. Let us suppose you are going to have protein family. Okay, let's suppose you are going to have a family of proteins, and let's suppose that protein family, okay, it is present in all the three domains of life. It is present in bacteria, it is present in archaea, it is present in eukarya. Then you can say that this protein family, it is well conserved. Okay, it is evolutionary conserved. Okay, so here you can see there are various enzymes of like glycolysis. Okay, if you are going to see glycolytic enzymes, they will be present in bacteria, archaea, eukarya. Okay, and when you are going to study the structure and function of those proteins enzymes, you will see similarity. Okay, so sometimes you can see the protein family, they might be present in various domains, and they might be evolutionary conserved. Now here, if you see the 3D structure of any of the protein, it is being stabilized by various factors. Okay, and hopefully you know the various factors which are going to stabilize the 3D structure. It can be hydrogen bonds between the peptide bonds, okay, between amino acids. What you are going to see, the presence of hydrogen bonds. Yes, ionic interaction, ionic bonds between amino acids which are going to have different charge. Yes, here van der Waals interaction, when two atoms they are going to come in close in contact, what you are going to see, van der Waals interaction. So hydrogen bonds, ionic bonds, van der Waals interaction, all these they can stabilize the 3D structure. But here if you see the disulfide linkage, a type of covalent bond. Okay, so hydrogen bonds, ionic bonds, van der Waals interaction, these are weak. But try to see disulfide linkage, a type of covalent bond, and generally it arises in between cysteine. So when within the proteins you are going to have amino acid cysteine. Okay, so what can happen? There can be formation of disulfide linkage. 2 cysteine can give rise to cysteine. Okay, so here disulfide linkage, they stabilize the 3D structure more, you can say, promptly. Now another very important thing which is going to stabilize the protein structure is hydrophobic interactions. Hydrophobic interaction, hopefully you know, okay, the polar, they attracts polar. Okay, in polar water you can dissolve, okay, polar things. So here hydrophobic interaction is in between hydrophobic things, like in between hydrophobic amino acids. So amino acids which are going to have hydrophobic R group, keep in mind they will try to cluster together, they will tend to cluster together. Okay, so here what is going to happen, due to hydrophobic interaction, the amino acid which are going to have hydrophobic R group, they will be present within the protein and they will be present together. Suppose you are going to have a cytosolic protein. Try to think here. Within the cytosol, you are going to have 70% water, hydrophilic environment. And let's suppose you are going to have a protein, and that protein is going to fold within the cytosol. Now keep in mind, this protein, which is a cytosolic protein, the hydrophilic amino acids, they will be present generally outside, while the hydrophobic amino acids, they will be present inside. Like attracts like. So here in this situation, keep in mind, the hydrophobic amino acids, they will be present inside, while the hydrophilic amino acids, they will be present outside. Why? Outside environment is hydrophilic. So like attracts like. So hydrophilic amino acids, they will be present outside, and the hydrophobic amino acids, they will be present inside. So here keep in mind, due to hydrophobic interaction, the amino acids with hydrophobic R group, they will try to cluster together. Okay, let us discuss another situation. In case of membrane proteins, you are going to have different situation. Try to recall the structure of the plasma membrane. Okay, try to see here. In the case of membrane proteins, here try to see the environment around the membrane protein. It is hydrophobic. So here hydrophobic amino acids, they will be present outside. So here in the case of membrane proteins, the hydrophobic amino acids, they will be present outside. Just try to recall the structure of membrane. Okay, try to see. So if you are going to have membrane protein, okay, it is present inside the membrane. So outside you are going to have hydrophobic environment. Okay, hydrophobic tails. Now try to see here. Here in the case of membrane proteins, the hydrophobic amino acids will be present outside, while the hydrophilic ones, they will be present inside. Different situation. Clear? let us move forward. Now try to see here. Now we are going to discuss the structure of a protein known as myoglobin. We are going to discuss the complete 3D structure of the protein myoglobin. Here, if you are going to see the structure of myoglobin, try to see here. Keep in mind, myoglobin, this protein is going to belong to a family of protein known as globin family. Here, keep in mind, hopefully you remember that there is another protein, hemoglobin, okay, that belong to the same family, globin family. And here try to recall the function of hemoglobin. The function of hemoglobin is to transport oxygen. Hopefully you remember, in RBC, red blood cells, what you are going to see? Presence of hemoglobin. Keep in mind, red blood cells, just they are a bag full of protein, hemoglobin. And what is the function of hemoglobin? To transport oxygen. Now, here, if you see the function of myoglobin, the function of myoglobin is to store oxygen. Myoglobin function is to store oxygen. What is the function of hemoglobin? To transport oxygen. Now, here, keep in mind, the myoglobin protein, this is present generally in the muscles, okay, and the function of this protein is to store oxygens. And the most important thing, this protein myoglobin is going to have higher affinity for oxygen than hemoglobin. Try to see here, hemoglobin, okay, when it is saturated with oxygen, okay, it has to transport the oxygen to muscles, okay. So within the muscles, you are going to have this myoglobin. Now this myoglobin is having higher affinity for oxygen. That's why it can, you can say, snatch oxygen from the hemoglobin. So here keep in mind, myoglobin, it is going to have higher affinity for oxygen than the hemoglobin. Now here if you see the structure of myoglobin, here I just want to tell you that in myoglobin, what you are going to see, you are going to see that it is going to have a single polypeptide chain. Here in the myoglobin, you are going to have single polypeptide. It is going to have 153 amino acid. Okay, so total number of amino acid present in the single chain, it is 153. Now here, if you are going to see this single polypeptide, it is going to fold, and here what you are going to see, the percent residue, if you are going to see, most of this 78% is alpha helix. So here if you break all these super secondary structures, so here what you are going to see, 78% is alpha helix and 0% is beta. Okay, so here keep in mind, in myoglobin you are going to see most of the polypeptide, they are going to fold in form of alpha helix. You are not going to see the beta sheets, beta structures. Okay, so here also keep in mind, in myoglobin what you are going to see, all the peptide bonds, they are going to lie in a single plane. So here all the peptide bonds which are present within the single polypeptide, they are going to lie in a single plane. That's why we are saying all peptide bonds here are planar. Now if you see in the myoglobin, what you are going to see, that here the same rule is going to apply. The hydrophobic amino acids, they will be present inside. Hydrophobic amino acid will be present inside. Hydrophilic amino acids, they will be present outside. Very simple. Okay. Another, here if you are going to see that the hydrogen bonds ionic bonds, van der Waals interactions, okay, then hydrophobic interactions, all interactions, they are going to stabilize the structure of myoglobin. Now here, if you see the most important point, within the myoglobin, you are going to see the presence of proline residue within the alpha helix. Generally, proline, it is not present within the alpha helix, but here the proline residue which are present in alpha helix, okay, they actually are going to create kink within the helix, okay, and these kinks actually help in tight packaging of the helix. Okay, so this is about the presence of proline residue in the alpha helix of myoglobin. Now let us discuss another important point related to the myoglobin. So here within the myoglobin you are going to see 6 ligands. Now here out of 6 ligands, the 4 ligands, okay, generally the 4, you can say here the nitrogen atoms, okay, they are going to make the ligand, and they are actually going to make a ring. That ring is porphyrin ring. Now here I just want to tell you the 5th ligand, okay, here it is the histidine amino acid, and this histidine, okay, you know very well, it is going to have imidazole group, and this imidazole group, it actually stabilizes the heme group. And here if you see the 6th ligand, it is going to interact with the oxygen, okay, it is actually the oxygen binding site. Don't worry, everything will be clear. Just have a look into this image. Try to see here. Here if you see in the center, you are going to have the iron, okay. Now here you are going to see 4 nitrogen, okay, 1, okay, 2nd and 3rd and the 4. So here the 4 ligand, okay, they are interacting with the iron. Now the 5th one, nitrogen from the histidine, okay, that is also interacting with the iron. And now above, what you are going to see, the oxygen, okay. So here you can see the presence of heme. Remember, it is a prosthetic group, okay, which is present within the hemoglobin. It is also present within the myoglobin, okay. So here iron has the capability to bind to oxygen. Now here keep in mind, in the myoglobin, okay, here if you see within the pocket of the myoglobin, you are going to have the prosthetic group, and this prosthetic group is heme, okay. So heme, keep in mind, it is present in myoglobin, it is also present within the hemoglobin. Now this group, prosthetic group, okay, here within the heme you can have iron. Now this iron can be present in 2 forms, ferrous or ferric, okay. Fe2 plus, it is ferrous. When the iron in the heme, it is present within the ferrous form, it will bind to oxygen. When the iron, it is present within as ferrous form, it will bind oxygen. When it is present in ferric form, remember, it doesn't bind to oxygen. So here keep in mind, so here the iron in one state, it has the capability to bind oxygen. In the another state, it doesn't bind oxygen. So this way the myoglobin, it can store oxygen, and whenever the cell need during heavy muscular contraction, okay, myoglobin can also release the oxygen, okay. Now hopefully all the points related to structure of myoglobin are clear to you. Now have discussion later to heme. So keep in mind, heme is responsible for deep red brown color of the hemoglobin. Okay, you know very well, if you see the color of the blood, okay, so here in RBC, what is there? It is presence of heme, and RBC, you know, they are going to have deep red brown color. Why? It is due to the presence of heme. Now here I also want to tell you, in animals like whales, seals, okay, they are having more, you can say, presence of myoglobin. In their muscles, you are going to have abundant myoglobin. And in myoglobin, you are going to see the presence of heme. So if you are going to see the muscles of animals like whales, seals, what you are going to see? You are going to see the brown color. Why it is there? Due to the presence of myoglobin, and within the myoglobin you are going to see the presence of heme. Now these animals, you know, whales, seals, they can go deep within the sea, and they can remain within the sea, okay, for a longer duration. So here keep in mind, their muscles, they have much higher capability of storing oxygen. Why? Because in their muscles you are going to have more presence of myoglobin. And hopefully you remember, what is the function of myoglobin? Myoglobin, it can store oxygen. So that's why they can go deep within the oceans, and they can remain within the oceans for a longer duration. Hopefully all the points are clear to you, and you have learned clearly about proteins 3D structure. Thank you for watching this. Please do like, share the video, subscribe to my channel Deep Learning Biology. If you have any doubt, please leave the comments. Okay, the most important thing, please revise. You have devoted your time in your brain. What you have done, you have framed new connections between neurons. Okay, now make those connections stronger. Just revise your notes. Okay, yes, you can watch this video again. You will feel more comfortable. Yes, before going for sleep, try to revise things. Very important. During the time of sleep, the brain is going to process memory. Things, they will move in your long-term memory. So, learn here that during the time of sleep, what is going to happen? The things, they will be going in your long-term memory. So, before the sleep, just, okay, in a relaxed manner, try to revise, try to recall whatever you have learned. Hopefully, in next few days, you will enjoy the process of learning. Bye bye. Take care.